Hello everyone, this is the Histology review for our extra credit super quiz coming up soon. There are a few things that you probably want to make sure that you need to know for each of the tissue types that I'm going to talk about today. You should A, know the name of the tissue type. B, you should know what type of fibers make up the tissue type, at least predominantly. Most type of tissues have more than one type of fiber that you'll find in them, but you should know what the main constituent is. That's more true for connective tissue than epithelial tissue. 3, you should know what type of cells you would predominantly find in that tissue type. And 4, you should know where in the body that type of tissue would be found. And of course, you should be able to recognize the tissue, which is what we're going to work on right now. So starting off, this is simple cuboidal epithelium, and there are a few ways to recognize that. First off, simple cuboidal epithelium is not typically going to be found just by itself. One of the first things that you have to ask yourself whenever you're looking at a tissue is, the first thing you should ask yourself is this epithelium or not epithelium? And then you can get into what type of epithelial tissue it might be. But remember that the epi part of epithelium means on top, right? Near the surface. One of the features of epithelial tissue is that it is pretty much always a coating or lining of some sort. That means that it's going to face an open area. That open area could be the outside world, which your skin faces, or it could be an internal cavity, something like a body cavity or the lumen of a blood vessel or the inner area of a tubule in the kidney, a nephron, something like that. But epithelial tissue will always face an open space on one of its sides. And you can see here that this area right there that is labeled free surface or lumen, that's empty space. So if the tissue that you're looking at does not border an empty space, it has like different types of tissue on each side of it, it's probably not epithelial tissue. Cuboidal epithelial tissue, as you would get from the name, is typically going to be about as long as it is wide. So here you see the height, the width, and each of these cells describes sort of basically a square. Epithelial tissue, all epithelial tissues are cell dense. That means that you have cells right up next to other cells with very little to no extracellular matrix in between them. So if I outline, the cells here, you can see that, pretty much each cell is right up next against another cell with no extracellular matrix, no fibrous tissue, nothing like that in between them. So that's a key identifying factor of epithelial tissue, is cells tightly packed with other cells. So we've talked about how to identify it as epithelial tissue, how to identify the cuboidal shape, and how do we know that this is simple rather than stratified, and because you do see more than one layer here, but what you have to look at is the epithelial tissue one continuous layer? So this right here that I'm outlining in red, that's a fibrous area. Now that's the basement membrane. The basement membrane is the bottom of a set of epithelial tissue. It goes from the free surface, the lumen, to the basement membrane. And you can see here that from the lumen to the basement membrane, that's just one cell. Now there are more cells up here, but they're on the other side of that basement membrane. So if your cell layers are separated by a layer of connective tissue, even if it's a thin layer of connective tissue, then they're not the same layer. So this is only one layer of cells thick. You have more cells up here. You have more cells of a different type down here. But this is simple cuboidal epithelium. There's one layer of cells from the lumen to the basement membrane. This is also simple cuboidal epithelium. As you see, this area here, that is the free surface or lumen. This area here, that is a basement membrane. That's a different type of cell. And these cells are tightly packed next to each other. Note that each cell has a nucleus and some cytoplasm. That's how you can tell it's a cell. I don't see a lot of extracellular matrix in between them, and they're roughly the same height as width. So this is simple cuboidal epithelium. This is also simple cuboidal epithelium. This is a configuration that you will often find in internal organs, particularly like the kidney and the liver, where you will find simple cuboidal epithelium that is arranged in tubes. With a basement membrane surrounding the tube, and then other tubes as well. And each of these tubes is going to be lined with a layer of simple cuboidal epithelium. This is stratified cuboidal epithelium, and remember that stratified just means more than one layer. And in this particular case, the epithelium is this area right here. All of this down here, that's actually connective tissue. So if we have an arrow that's pointing there, that would be stratified cuboidal. If we have an arrow that is pointing in there, that is, looks to me like dense irregular connective tissue. We'll get to that in a second. Now, how can you identify this as stratified cuboidal epithelium? Well, if we zoom in here just a little bit, and you can see that each of these is a cell with a nucleus. The height is about the same as the width. And there are two layers of cells. Now, 2 layers isn't very many layers, but it's still more than one. And since we have 2 layers on top of each other with no connective tissue in between them, that makes it stratified. Cuboidal epithelium, when it is stratified, is often stratified with only 2 layers. That's a feature that you find in many glandular tissues. This is stratified cuboidal epithelium that has more than 2 layers. And you can tell that it's epithelial tissue, or at least this stuff here in the purple is epithelial tissue. And you can tell that because of the dense packing of the cells. So this is a cell, this is a cell, this is a cell. This down here is a cell, that's a cell right there. Here we have a cell. A bunch of cells packed close together with no space, no extracellular matrix, no fibers in between them. These are relatively cuboidal cells because if you look at them, they aren't column-shaped. They aren't much higher than they are wide. They aren't squamous. They're not much flatter, much shorter than they are wide. Their height and their width is about equal-ish. They're not exactly square. Some of these have, more than 4 sides, but they still have about equal height and width. So these would be cuboidal, and we can see that there's definitely more than 1 layer. Now you can see there's a few cells here that look like they're kind of flat. That one looks like it's kind of flat. It is not uncommon in sections. You might catch just the edge of a cell, or you might have some cells that are sort of deformed. Just because you see one flat cell, that doesn't mean that, oh, it's squamous, or, now you can't tell what it is. You should use your judgment when you're looking at tissues and say, what is the predominant feature here? What is the feature that I see most of the time, understanding that biology is a sloppy process and very few things are going to be 100%. This is a simple columnar tissue, and you can tell simple columnar. So here, this right here is the basement membrane going across there. That's what's going to separate the epithelial tissue from the connective tissue. These are epithelial cells. You can tell that because they are closely packed together. There's very little extracellular matrix or fibers surrounding them. Each one has a nucleus and some cytoplasm, and they're pretty clearly column-shaped, rectangular. Most, not all, but most columnar epithelia will have the nuclei located towards the bottom. And this is simple. We see nice squares for each of them, or nice rectangles for each of them at least. They aren't overlapping each other with nuclei out of place. There's not more than 1 layer, so these are a simple layer of columnar tissue. This area down here is some sort of connective tissue. This is pseudo-stratified columnar epithelium, and it can be a little bit difficult to tell the difference between pseudo-stratified and simple columnar. You have to pay attention to the shape of the cells and look at, are we getting cells that are going all the way down in nice columns with a single row of nuclei in them? Or do you have sort of multiple rows of nuclei where you can't necessarily see all of the columns lined up in neat rows? All of these are columnar cells. They are cells that all of them are touching the basement membrane down here, and all of them are touching the lumen up top, you can tell that this is epithelial tissue, by the way, because densely packed cells, and they're facing an empty lumen area over here. But due to the way that the cells grow, they don't grow in nice even lines. They kind of like intertwine with one another. So that means that in the section, you can't always see an entire column growing by itself. And so you'll see these nuclei kind of out of place as the cell comes into and out of the frame of the section. And so you don't really see a nice even line of nuclei towards the bottom. If you outline the cells, you don't see nice even rectangular cells. That's what tells you that it's pseudo-stratified. Pseudo-stratified cells are also commonly, but not always, ciliated at the top. Cilia and microvilli can be difficult to tell apart. Cilia are larger, easier to see on a light microscope, and often associated with pseudo-stratified columnar. So this is stratified squamous epithelium, right? So how can we tell that it is epithelium? Well, we are facing an open luminal area, right? It is epithelial tissue. We can tell because all of these cells, and you can tell that they're cells because each one has a nuclei, they're all densely packed. We don't see a lot of connective tissue fibers. Each of these outlines is a cell, with a nuclei. Most of them have nuclei in them. Some of them, again, you're not going to be able to see the nuclei on because maybe it wasn't in the part that got sectioned. But if you look at the tissue overall, you can see that each of these cells is packed closely to the other cells, and by and large, most of them have nuclei, and it's very densely packed with very few fibers. We can tell that it's stratified, clearly, because there's lots of layers. That's not really a question. And as for the shape, well, if you look down here, you will actually see a few more that are kind of a little bit more cuboidally shaped. But the predominant shape in this section is contained in this area up here, and those are all flat, sort of fried egg shapes. You can tell that this is actually living stratified squamous epithelium as opposed to dead keratinized squamous epithelium, because even here at the top, this area that's facing the lumen, these cells are still, they're not hollow, and most of them still have their nuclei inside. That would tend to indicate that they're living cells, not dead keratinized cells. These are dead keratinized or keratinized stratified squamous epithelium, at least these up here on the top are. Down here, this is the stratum granulosum, stratum spinosum, and basale down here, and these are clearly live cells. Now, dead stratified keratinized squamous epithelial cells are something that you are pretty exclusively going to find in the skin. And you guys have all studied skin. You know, generally speaking, what skin looks like. So one of the first things that you should ask yourself is, does this look like skin? And if so, are these dead keratinized cells on the top? And so you can tell, if you look down here in the layers down below, you'll see some more cuboidal-looking cells, and then they transition to being squamous cells here. And each of these cells down here has a nuclei in it. If you look at these cells at the top, you'll see they look fairly hollow, and you don't see any nuclei in them. It is important to pay attention to the fact that these are cells. right? This is not connective tissue. We're not looking at fibers here. The dead keratinized layer of your skin can look like some connective tissues, and we'll talk about those later, but you should learn to recognize them in the context that they're likely to appear in a body. So you would never see connective tissue that is facing a lumen or the outside world. You won't see it in association with these living cells underneath it. It won't look like it's on a piece of skin. All right, into the connective tissues. Areolar connective tissue is a loose connective tissue. You can tell areolar tissue by a medium dense amount of cells. There's a variety of different cells here, but probably the most common is going to be fibroblasts. And so you have, a relatively medium to medium high density of cell tissue, and you also see a bunch of different types of fibers. These big, thick, pink fibers here, those are the collagen fibers, and you should see, mostly these big, thick collagen fibers. But the thin fibers, like that one there, this one here, this one here, those are elastic fibers and or reticular fibers. Most of them are probably elastic fibers. So you should see a mix of different fiber types. You can tell elastic fibers because they're thin and they're usually darker. You can tell that this isn't epithelial tissue because there's no luminal surface that it's facing. The cells are not packed tightly together, and you see the presence of these fibers running in every which way. And with areolar tissue, you should see, first off, that the fibers are going in different directions. They're not all parallel. And secondly, that you'll see empty spaces in there, and that you'll see multiple different types of fibers. Remember that the predominant type of cell in areolar tissue is going to be fibroblasts. The predominant type of fiber is going to be collagen fibers. Here's more areolar tissue, and again, we can see these big, thick, pink collagen tissues, the thinner, darker, elastic tissues, and some reticular fibers as well. Reticular fibers can look like elastic fibers, but they are more branched. And lots of cells, but they're not densely packed with each other. Most of these cells are going to be fibroblasts, but we have a few of them. This one here, this is a mast cell. Here are some blood cells. There are lots of different types of cells that move through areolar tissue, but it is made by predominantly the fibroblast cells. So this is adipose tissue. Adipose tissue is very easy to recognize. What you're going to see with adipose tissue is largely, large, bulbous, empty cells with nuclei that have been pushed over to the side of them. This large empty area, that's the fat storage globule, and fat does not usually stain very well, so it looks empty because it's unstained. The nucleus does stain well, and it's usually going to be pushed to the side. So you're going to be looking for large cells that kind of look like foamy bubbles. The cells will be relatively densely packed together, but because the cells are so big, the nuclei will be spaced fairly far apart from one another. You can tell it's not epithelial tissue because, first off, there's no epithelial tissue that looks like these type of cells. And secondly, adipose tissue or fat tissue usually will not be facing directly to a lumen or open area. The type of cell that you're going to find in adipose tissue are adipocytes, or fat cells, and they're not a very fibrous tissue, but the type of fibers that there are that hold these fat cells together are actually reticular fibers, though that's probably not something that I would ask you on a test. Reticular tissue, all right? Reticular tissue is a loose connective tissue with a dominant fiber type of reticular fibers. Reticular fibers usually show up on a histology stain as being thin, dark fibers that are highly branched and sort of follow all of these branches. Reticular tissue is found mostly in the lymph nodes, as well as in some associated organs, the liver, things like that. And what reticular tissue is really good at is holding a bunch of cells together while still allowing them to freely move, right? So in your lymph nodes, you have a lot of particularly blood cells, white blood cells, that are always moving in and moving out of your lymphatic tissue, and they have to be able to be held there at a high density, but also be able to move in and out of that tissue. They're not permanent residents. So the features that you're going to look for to identify reticular tissue is a relatively high cell density, first off. So we see cells here, here. If you look around for the nuclei of cells, you see a lot of cell nuclei. Most of these in this case, because this is a lymph node section, I believe, are going to be blood cells. They're going to be fairly densely packed, but you will still be able to see fibers, these dark reticular fibers that are branched in the background. It will not usually be a tissue that faces a lumen, so you can distinguish it from epithelial tissue by the presence of dark fibers, as well as by the section not facing any sort of lumen or open area. The type of cell that makes reticular tissue is a specialized type of fibroblast, but in lymph nodes, which is the most common place to find reticular tissue, most of the cells that you're going to see are going to be white blood cells of some sort. The predominant fiber is, of course, reticular fibers. Here's more reticular tissue, and you can see again the presence of lots of different cells, fairly densely packed. But you can also see these dark branched fibers that are the reticular fibers, and they form a web or net that are sort of like holding the cells in place. Reticular fibers have lots of areas for cells to sort of grab onto. All right, dense regular connective tissue. So the features of dense regular connective tissue is lots of thick, usually, though not always pink, depends on the stain that's used, collagen fibers. Collagen fibers are the dominant tissue type in dense regular connective tissue, and they will all be moving the same direction. So you can see the grain of these are all sort of moving the same way. Another thing that you will see is dispersed nuclei that are relatively flat. All right. So here you can see this is a nuclei here, this is a nuclei there, this is a nuclei, this is a nuclei, this is a nuclei. But if you draw a circle around the cells, and these cells aren't actually much bigger than the nuclei, then you'll see that the cells themselves are not densely packed. This is a distinguishing feature of dense regular connective tissue, is that you have relatively dispersed cells. Most of the tissue is collagen fibers, extracellular matrix, not cell mass. The cells are mostly there to just lay down the collagen fibers. And the cells and the nuclei will both be flat shaped so that they don't interfere with the tight packing of all of these dense collagen fibers. Dense regular connective tissue was commonly confused with stratified squamous cells on the test. And I can sort of understand why, because they're both going to have kind of parallel stacks. They're both going to have sort of flattish cells. But here is the way that you can distinguish them from each other. First off, squamous epithelial tissue as an epithelial tissue will almost always face a lumen or open area on one side. Secondly, stratified squamous epithelium is usually going to be seen in the context of skin or some sort of like lining of your mouth or something like that. So it will be associated in a context, which is layers of squamous cells on top of some, basal cells, and then on top of dense irregular connective tissue, dermis underneath. So you can sort of recognize it from the context. You can also recognize it because if we outline the cells here, you will see that the cells tend towards being flat, but they're densely packed with each other, and each cell has a sort of round nucleus in it. If we look at the dense regular connective tissue, what we can see is that the nuclei, the dark stained portion, is flat, but they're dispersed. You don't see a bunch of these next to each other. They're not densely packed with each other. Most of it is this fibrous tissue that is collagen fibers that's going down. In stratified squamous cells, you're going to have each cell next to another cell and a fairly dense pack of nuclei together. Whereas here we will see interspersed nuclei with mostly lots of fibrous tissue going down. Dense irregular connective tissue has, again, thick collagen fibers, but instead of all going the same direction, they're going to be going kind of at random directions and squiggling, and some of them will be coming out at you, so you'll see a cross-section of them. Some of them will be longitudinal, but you're still going to see mostly collagen fibers, and the nuclei of the cells, which should be fairly visible, will be just sort of interspersed, not very dense. So you can see that there aren't actually a whole lot of cells here. These nuclei are going to be much less flat than we saw in dense regular connective tissue because the fibers are going every which way, so you don't have to flatten out to fit in between the tightly packed fibers. The fibers themselves will be going a bunch of different directions, running around and underneath each other, and you will occasionally see these open areas where just like all of the fibers zig instead of zag, and you get sort of an open area. Dense irregular connective tissue will also sometimes have blood vessels or capillaries with epithelial tissue running through it, and you can sometimes see those. Cartilages. So there are a few things to keep in mind when you are identifying cartilages. All right. All cartilages will have chondrocytes, that's the type of cell, that is a cartilage-making cell, located in lacuna. So here we see this is a chondrocyte right there. This is a chondrocyte right there. This is a chondrocyte right there. And the sort of open, empty space around them, that is the lacuna. Hyalin cartilage is identifiable because the background, the matrix between the cells, is going to be fairly non-fibrous and sort of translucent. It'll look kind of like, you know, Jello or something like that. You have cells that are, this is clearly not epithelial tissue because the cells are not densely packed. There's extracellular matrix between them. We are not facing some sort of luminal or open area. And we have a medium to medium dense packing of the cells here, and they're relatively spread out fairly evenly. Fibrocartilage is identifiable because, again, we have our chondrocytes, and many of those chondrocytes that you can see are in lacunae, these open rooms. But we also see fibrous, usually though not always, pink collagen fibers running relatively parallel. In fibrocartilage, the nuclei are usually more oval shape and less flat because they have these lacuna rooms to sort of expand into. And you will also often see sort of the cells clustered together. So the cells in fibrocartilage are fairly dispersed. You don't see as many of them, but they tend to cluster up. And they tend to cluster up frequently, though not always, in lines. So you can see here's a cell, 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 and they're all running down that line. That has to do with how they reproduce around the collagen fibers. The collagen fibers tend to pack all of these related cells together in the same sort of line area between the fibers. So look for these lines of chondrocytes in lacunae to identify fibrocartilage. But if you see, you know, big, strong collagen fibers with chondrocytes in lacuna, it's probably fibrocartilage. Elastic cartilage. In elastic cartilage, what you usually see is larger cells in larger lacunae. So here we have, this is the nuclei of a cell. This is the lacuna. And they are going to be fairly densely packed. So you'll have lots of cells, big cells, in lacuna, but you can tell that it's not epithelial tissue because there is still extracellular matrix between the cells. And that extracellular matrix will have fibers in it, in this case, mostly elastic fibers, which are thinner and darker than collagen fibers. Elastic cartilage will also typically not face a lumen or open area. So fibrocartilage versus dense regular connective tissue. This was commonly confused on the test. How can you tell these two things apart? Well, they are kind of difficult because they do both have fibers, collagen fibers that are running parallel to each other. Here you can see the lacuna. Yeah, blue on blue doesn't work very well. You can see the lacuna here with the cells inside, and you can see how the cell nuclei are sort of ovoid, not these flat, long lines. Whereas over on the dense regular connective tissue, you can see sort of these flat nuclei interspersed, and that they're definitely not in lacuna. And that's probably the main way that you're going to tell them apart. You're going to look at the shape of the nuclei. You're going to look at, do these nuclei tend to be surrounded by a lacuna? Elastic cartilage and stratified cuboidal are also often confused, and there's an easy way to tell them apart. First off, stratified cuboidal epithelium are going to typically face a lumen. You can see the luminal facing here. Secondly, the elastic cartilage, even though the cells are fairly tightly packed, you can see the extracellular matrix between them, and you can usually see the fibers in the extracellular matrix, which indicates that even though they're packed closely together, they are connective tissue rather than epithelial tissue. And last, you can tell by the typical lacuna with chondrocytes inside of them in the elastic cartilage tissue. In the epithelial tissue, you will see the cells do usually have a nucleus inside of like a cellular area, but it looks very different from this sort of very clear lacuna and chondrocyte motif. If nothing else, the stratified cuboidal epithelium, the cytoplasm of the cells, usually stains pinkish, light pinkish, whereas the lacuna outside of the cellular area in cartilaginous tissue is usually staining clear. Distinguishing between the three types of cartilage can also be tricky. I just want to reiterate some of the differences between them. In hyaline cartilage, what you'll see is an extracellular matrix that is sort of translucent and usually non-fibrous. You'll see a kind of random dispersion pattern of chondrocytes in lacunae that are medium to medium dense or to medium high density. In elastic cartilage, you will have highly dense cells with the the ECM in between them will have sort of thin black elastic fibers in it. The cells will usually be larger. In fibrocartilage, what you will see is dense, thick, parallel cartilaginous fibers. You will see your chondrocytes in lacuna that typically occur in kind of lines together. So there will be fewer of them than in either hyaline or elastic cartilage, but they'll be kind of bunched up together. So you see a non-random spotty dispersion pattern there. All right, so for this next part, I'm going to show you guys some slides. I'm going to give you a few seconds at the beginning of each slide to pause the video and see if you can determine what type of tissue it is. And then I'm going to reveal the tissue type and tell you how you can figure that out. So go ahead, pause the video now, and figure out what type of tissue this is. All right, so this type of tissue is adipose tissue. I hope everyone got that. You can tell the adipose tissue because of the large empty cells with the fat globules in them, sort of densely packed, and with the nuclei pushed off towards the side. All right, go ahead, pause, figure out what type of tissue this is. Okay, so this is pseudo-stratified columnar epithelium, and you can tell that, first off, it's epithelium. You can see up here there is a ciliated border, so you know that it's facing a lumen, but you can also see the lumen up here. They're definitely columnar cells. They're not cuboidal or squamous. And if you line them out, you can see that they aren't making a simple layer of rectangular cells. They're sort of overlapping each other, going in and out of the field of view. And they're not making a single, nice, even layer of the nuclei. The nuclei are sort of pushed into multiple rows. So that indicates pseudo-stratified. All right, here you go. So this is dense, regular connective tissue. You can see the flattened nuclei of the fibroblasts, and you can see the collagen fibers running in parallel bundles. So for this one, look at the tissue type that the arrow is pointing at. Okay, this one is simple cuboidal. So if you look here, you will see that these cells here, where the arrow is pointing at, are facing into this inner area, which is empty. That's a lumen. And each of these cells is densely packed next to the cells next to it. Each of them is about as high as it is wide, at least in general. And you can see that underneath it, what we see is a different type of area. This is not more cuboidal cells. This is some sort of connective tissue underneath it. So we have one layer of cuboidal cells facing a lumen densely packed with each other, simple cuboidal. All right, so this is elastic tissue. Now, I don't actually think I might have forgotten to talk about elastic tissue earlier on. I might go back and try to edit that in at some point. But elastic tissue is different from elastic cartilage. Elastic cartilage is cartilage. It is chondrocytes in lacunae. Elastic tissue is going to be found in some of the stretchy tissue of the heart, the aorta, and it is a dense, regular connective tissue, but instead of being a collagen-based dense regular connective tissue, it has lots of elastic fibers. And so you can see these black fibers are not collagen fibers. Collagen fibers are thick and usually stain red to pinkish. These are sort of squiggly and dark black. I can tell that this fiber is not currently being stretched, so part of what makes these fibers elastic is that as this fiber was to stretch, or as this tissue was to stretch, these fibers would straighten out and become straight parallel bundles. And then as it relaxes, they kind of coil back up. This is a dense tissue, you can tell, because you've got lots of layers of these fibers on top of each other. You can distinguish it from reticular tissue because reticular tissue is not going to have the fibers in parallel bundles. There will be fewer fibers and more cells in reticular tissue. And you can see here that there are some cell nuclei relatively flat and interspersed. And those are mostly going to be fibroblasts. So in this tissue, the type of cell that you would find is fibroblasts. The type of fiber that you find is predominantly elastic fiber. All right, here we go. Okay, so this is areolar tissue. You can tell because you have thick bundles of collagen fibers. You have these thin, dark, thin, dark, elastic fibers. And you have sort of medium to medium dense dispersal of cells, mostly fibroblasts, but you still have open areas here, 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 which are areas that cells can use to move in and out. Okay, so this is dead keratinized stratified squamous epithelial. We're talking about up here where the arrow is pointing. All right. So these are, you can sort of tell from the context that this looks like skin. You have dense regular connective tissue down here. You have living epithelial tissue up here. And then at the top, you have these cells that are empty and that don't have any nuclei in them, indicating that they're dead cells. And of course, it's facing this luminal empty area, indicating that it's epithelial tissue. And really the only top layer of dead cells that you'll have facing a lumen is usually going to be dead keratinized stratified squamous epithelium. Okay, so let's do the reveal. This is reticular tissue. And so you can tell that because the fibers in it, which you can see, are dark and thin and highly branched, not all running in parallel bundles. And there is a relatively dense population of cells being held in place. Most of these are going to be white blood cells, but not all of them. And so this is reticular tissue, probably from a lymph node. All right, let's do the reveal here. So this is Hyalin Cartilage. You can see that the background is non-fibrous, So you look at this background, you don't really see any fibers running through it. Smooth, sort of jello-like. These are clearly chondrocytes in lacuna. Have that very typical shape to those. And you can see that the cells are sort of dispersed in a medium dense pattern all around. They're not all bunched up into lines. You don't see any elastic fibers in the background. All right, and last one. This is Fibrocartilage. You can tell because of the thick collagen fibers running parallel. The chondrocytes in lacuna, let's see here, that have sort of rounder, kind of egg-shaped nuclei in their space, but they are held into these sort of linear bundles, right, where the cells tend to cluster in lines in between the collagen fibers. All right, so that completes our review of histology identification. I hope that you have found this very useful, and I apologize for sort of runny nose. I think that I'm starting to come down with something a little bit. But I hope you guys all get a full 20 points on the extra credit histology quiz, and I will see you in class.